Okay, it just hit 1:30 on my watch, UK time. Hello, everyone. Good afternoon, good evening, good morning, depending on where you are joining us from across the world. Thank you so much for joining us, and welcome to yet another edition of the GSU Malaria Journal Club, which is a monthly series. Today's webinar is a parasite-focused one, referencing Nina White and Chiyun Lee's recent preprint as well as app titled Pf-HaploAtlas, an interactive web app for spatiotemporal analysis of Plasmodium falciparum genes. For those of you that do not know me, my name is Pf-HaploAtlas, and I'm a training coordinator at Malaria Gen, working with a team of awesome people to organize these monthly events. Today's going to be slightly different, but also exciting in how we run our journal club today. There will be a presentation part of it, as well as a demonstration. And we have two speakers instead of one. Just a few ground rules. The journal club is being recorded and will be available on our YouTube channel later today, the Malaria Gen YouTube channel. So please do check it out. Share around on your social media, Twitter, LinkedIn, and with your networks and colleagues. Kindly stay on mute so that we don't disturb our speakers. As I said, we will have a Q&A session, as well as bring your own gene demonstration at the end, where you can raise your hand or type your question and comments in the chat or Q&A box. And again, I repeat, please raise your hand and unmute yourself to engage further with both our presenters. Without further ado, I would like to introduce both our speakers for today. Dr. Nina White and Chiyun Lee, who are based in the parasite team within the GSU. So GSU Strong. The title of the presentation is similar to the HaploAtlas app and paper that you are gonna go through. I'm gonna read a short bios for both of our speakers. I'll start with Dr. Nina White, who's a senior data scientist within the GSU. Nina specialized in molecular ecology as well as conservation genetics during her bachelor's degree and PhD. For her PhD, she focused on the co-evolution of a critically endangered amphibian and a fungal pathogen. Throughout her career, she has used genomic data to tackle infectious diseases. Currently, Nina is a senior data scientist in the malaria parasite surveillance team at the Genomic Surveillance Unit, Wellcome Sanger Institute, where she processes, analyzes, interprets genomic information for malaria parasites, and develops tools to make these tasks more accessible for the wider community. Next, I'll introduce Chiyun Lee, who's a senior data scientist within the GSU at the Wellcome Sanger Institute. Chiyun studied biochemistry and pathology at university, where he gained research experience on malaria transmission blocking vaccines and repurposing drugs for tuberculosis. He then joined AstraZeneca to do research on omics-based drug discovery methods and machine learning. Chiyun is currently a senior data scientist in the malaria parasite surveillance team and at the GSU within the Wellcome Sanger Institute, where he develops new software, data pipelines, novel algorithms, and apps for the malaria community. So true to their bios, welcome to our speakers. Without further ado, please go ahead and give your presentation as well as demonstration. Chiyun, go ahead.
Awesome. Thank you for introducing us, Babalo. And hi, everyone. Thank you for coming to this journal club session. Nina and I are really excited to be able to present to you the HaploAtlas. It's a project we've been working on for quite some time now. So it's really exciting that finally the day has come for us to be able to share this more widely with the Malaria Gen community. So yeah, should we get started? So on the next slide, we will see the contents of the talk. So again, as Pabalo mentioned, this will be quite an interactive session for a journal club, as we'll be trying to get the community to learn how to use the HaploAtlas through both the live demo and an audience participation section. And here we have these sort of contents of the talk. So first of all, I'll be sort of speaking a little bit about why the HaploAtlas was developed in the first place, giving a little bit of background for roughly 10 minutes. And then we'll move on to Nina, who will be showing you the sort of demo of the app. and we'll cut to question and answering. And then finally, with the remaining time we have in the session, we'll do the live analysis. So do be prepared to raise your hand, and once Pablo selects you, be ready to sort of speak with me and Nina about what your favorite gene is, why it's your favorite gene, and then sort of as three, we can see what the Pf-HaploAtlas tells us about that gene that you've selected. Okay, so let's get started then. So first of all, why was the Pf-HaploAtlas developed? So to answer that question, on the next slide, we'll see we need to first talk about the Malaria Gen Pf7 dataset, which is, as we all know, a very key resource for genomic surveillance in the malaria community. Well, it holds 20,864 Plasmodium falciparum whole genome sequencing samples, which is enormous. And what's really good about this dataset is that it's spread all across the world, really, across 33 countries, and it's literally decades of data. And that's thanks to all of the many study partners that have contributed samples to the Malaria Gen Pf7 data release. So yeah, big thank you goes out to everyone. Yeah, it's, this data set is enormous. It's literally terabytes, tens of terabytes in size, making it a little bit complicated to work with sometimes, but it does make it the biggest and sort of most expansive eukaryotic whole genome sequencing dataset of its kind. And what's really good about this dataset is that it's fully open, in that anyone can access the data, and we also have sort of fully open source Python packages and notebooks for accessing this data. And yeah, it's been fantastic to see all the different work that's been conducted using this data set by researchers all over the world, such as highlighting emergence of strains or parasite subpopulations, and analyzing drug resistance, this kind of thing. So if we move on to the next slide. So we do provide access to the data via Python package and Python notebooks, but I'm sure you'd agree it would be really convenient if you can sort of access the power of Pf7 with just a couple of button clicks on the internet, and you don't have to worry about how to work with gigabytes of BAM files and VCF files. So yeah, it'd be great if you can get an instantly accessible snapshot of evolutionary trends. And so that's the sort of idea we're trying to roll with when developing the Pf-HaploAtlas. So for example, you think of a gene that you want to quickly access, so you go into Google, go into the Pf-HaploAtlas, click around a few times, and then within a matter of half a minute, you realize you've analyzed several handful of genes. That's the sort of dream we're going for. And on the screen here, borrowing from a little bit from sort of software engineering principles, this is the sort of rough design specification that we are sort of working towards. So we wanted to build a tool which gives the user instant access of the evolutionary trends of all of the genes in the genome. And the sort of interesting problem here is that you don't want to be showing everyone all the sort of nucleotide SNPs in the genome, because that'll be way too much information and hard to draw insights. So we actually went for amino acid haplotypes because it gives you a nice balance of sort of biological significance without completely overwhelming you with tons of information. Next up, it's important that anyone can use the app and draw useful insights. So we really try to make the app friendly for both researchers and public health professionals for getting useful information from the app. Additionally, we wanted anyone to be able to access the app. So as I said previously, it'll be really good if we can make it sort of internet browser based, so anyone with a internet connection can achieve the same thing as someone with extremely fast internet with enormous computing clusters. And additionally, we want to minimize any computational demand required for people to access the data from the Pf-HaploAtlas. So really, we want people to get things on demand instantly. And then finally, we also want to sort of share this data as well. We want people to be able to build on top of what we've built. So actually, people can export the underlying haplotype data so that you can use it for your own personal needs. So that's it in terms of sort of rough overview. I'll hand over to Nina, who will take you through the demo.
Thank you, Chiyun, for that nice introduction. So now it's my turn to walk us through the Pf-HaploAtlas app. And I'm going to hopefully highlight for you some key features and outputs of the app and some interesting use cases as well. So here is the app. And the first thing that we need to do to get started is to pick a gene. So we come here to this search box, and you could scroll through a list of over 5,000 genes and pick one at random if you wanted. But if you have a gene in mind, you can also type it in. So to get started, we're going to look at the gene Couch13. So Couch13 is a very well-known gene. Mutations in this gene are associated with partial resistance to artemisinin and its derivatives, which are the current first-line treatment for malaria. So the first thing that Pf-HaploAtlas produces is what we term a haplotype upset plot. And this is the first of 3 plots that will be generated. There's some information here to help you interpret it, but I'll talk, for the purposes of the demo, I'll be talking through it. So there are 3 panels to the upset plot, and combined together, they give you an overview of the gene and all the haplotypes contained within it. So the first panel gives us a simple overview of the number of samples contained within each haplotype. An important thing to know about this plot is that haplotypes are displayed vertically, and each of the 3 panels display different information about each haplotype. We ordered them from the most frequent haplotype found to the least frequent. The second panel gives you an idea of the geographic distribution of all of the haplotypes. So we split this into the same populations as the PF7 data release and color code the samples accordingly. And over here in the sidebar, there is some more information on which color and abbreviations refer to which populations. And then finally, we have the mutational makeup of each haplotype. So dots indicate a mutation. So hovering over these information, we can see that this mutation here is C580Y, meaning that at the 580th amino acid position, a cysteine has changed to a tyrosine. So if we were interested in this C580Y haplotype, we can click on any of these 3 data elements relating to this haplotype and sort of dig down further with the next 2 plots that are generated by Pf-HaploAtlas. So you might have spotted there, we had a prompt to scroll down. And here we have the second plot, which is generated for a specific haplotype within a gene. So here, C580Y. And the idea of the abacus plot is to give you an overview of the haplotype's frequency through time and space. So we have a list of locations within countries here on the y-axis, and then a range of years on the x-axis. An important component of the abacus plot are what we call beads. These are these circles here, and beads indicate that samples were collected in a given location and year. The beads are colored, so and the color corresponds to the haplotype's frequency. So a gray cross indicates that no samples collected contained the haplotype of interest, and a black circle with a 100 icon means that every single sample had that haplotype. And an absence of a bead means that no samples were collected in that location and year. And what we really look for with the abacus plot are changing gradients of color across these beads as we go through time from left to right. So here, C580Y, we can see that there are some changing color gradients. Here in Thailand, no samples contained C580Y in 2001, But by the time we got to 2013, over half of samples did. Similarly, we can see in Vietnam, the frequency increased from 3% in 2010 to over 96% in 2018. So this tells us that there's something going on with this haplotype. And we do know that C580Y is associated with artemisinin resistance and is the most common artemisinin resistance associated mutation, definitely in Southeast Asia. So we'll move on from the abacus plot now, and we have another plot which relates to this specific haplotype, the world map plot. So the world map plot is sort of similar to the abacus plot in that it shows haplotype frequency across space and time. There are some differences. So you can choose a range of years here by changing the sliders, and you can customize that according to your needs. And this time we've aggregated the data on a country level. But same again with the abacus plot. The color intensity of the beads relates to the haplotype frequency. And this time as well, the outline of the beads corresponds to the population color. And to note, you can download any of these plots in various file formats by clicking on any of these orange buttons here. So that I showed you an example of an increasing haplotype frequency, but just quickly while we're looking at CELT, I'll show you what it looks like if the haplotype is decreasing. So here I'm going to select the wild type. When we hover over this haplotype, and it's a haplotype with no mutations, we can see the name of several lab strains which are identical to these haplotypes. So we know this one is the wild type or 3D7 reference strain. And just to show you quickly, here in Southeast Asia, we actually see the color gradient going the opposite way in the same locations. So the wild type decreased from 61% in 2010 to around 3% in 2018. So I'll just point out some other features and ways in which you can get more information or customize these plots. So the default minimum number of samples used for analysis is 25, but you can increase or decrease this depending on what you would like to do. Certain samples will have been filtered out. So we exclude samples with missing genotype calls or heterozygous calls, for example. And you just get some statistics here on those numbers. And these numbers will change between genes. Something important that Chiyun has already touched on is that Pf-HaploAtlas, as well as being an app that provides you these visualizations, it also acts as a database of analysis-ready data. So you can download the underlying data in different aggregations at the sample level or population level by clicking on one of these buttons here. And finally, you can also choose to view the sample count panel of the upset plot with the y-axis on a log scale, if you wished. You can continue to explore any gene in Plasmodium by clicking this button here. So that's a roundup of a very well-known gene in Pf-HaploAtlas and what Pf-HaploAtlas can show you. But a really important and quite exciting opportunity presented by Pf-HaploAtlas is the chance to look at lesser studied genes. Because we've used whole genome data as an input for this, we have information on all of the genes in the core genome, and they are there just waiting to be analysed. So we're going to switch to a gene called AAT1, and I can just type it in the search bar. AAT1 has only recently been associated with resistance to chloroquine, and because of this recent discovery, the gene is less well studied in comparison to a gene such as CELT. So again, we've generated the upset plot, and we're going to look at the second haplotype here. So we can see it was collected in 1,855 samples. It's predominantly found in the eastern part of Southeast Asia, this dark blue color. And we can see that there are two mutations making up this haplotype. So F313S and Q454E. If we click on this haplotype, I can show you that with the abacus plot, we see increasing color gradients as we go through time here in Vietnam. And this indicates to us that there's potentially some evolutionary pressure acting to select for this haplotype and raise its frequency in these locations. Finally, to just wrap up the demo, I'm going to take the opportunity to show you some examples of very diverse genes. Excuse me, I want to look at CSP. So CSP is of interest as a vaccine target. And you can see here how many haplotypes are contained within CSP. We can go even further by reducing the minimum number of samples to 5 instead of 25. And I'm sure you'll agree that this plot now looks very different to before, especially in the mutation makeup. And then finally, I'll show you an example of a gene which doesn't have much variation and what that looks like in Pf-HaploAtlas. So LDH is the protein encoded by the gene LDH is used in the main rapid diagnostic test, which doesn't use HRP. So scrolling down, we can see there are only 2 haplotypes contained within this gene. And if we go as far as possible and put the minimum number of samples to 1, there are still only 14 haplotypes contained within this gene, and the majority are only found in 1 or 2 samples. So that concludes this demonstration of Pf-HaploAtlas. I hope you found it useful, and I'm going to hand back over to Chiyun now to continue the presentation.
Cool. Thanks, Nina. So yeah, just to wrap up, before we break away for the question and answering, we just wanted to tell you about our next steps. So as mentioned already, we have a preprint out in bioRxiv, and we are currently in the process of submitting to a journal, an actual journal. And yeah, in terms of sort of the app development side of things, we will be updating the app once PF8 is released, which means that we're going to go from 16,000 samples to much higher, with more regions and more recent samples as well. And also, we'll be continuing to develop additional features to the app as well. And yeah, at this stage, we'd like to just sort of ask for the community's help, really, in suggesting what features to implement next. We'd like to learn what sort of use cases you'd like, how you're using the app. And you can do this by going to our GitHub page, where you will see a list of issues, a list of ideas that we already have and we're already testing. And yeah, you can go there and sort of suggest to us which ones you might like to see next. So yeah, and then on the next slide, yeah, and so many of you have already seen the amazing sort of Malarian article that the comms team has produced. But yeah, if you go here, you'll be able to find all the information. You'll be able to find the app yourself and have a play. You'll be able to see the link to the bioRxiv preprint and have a read about the sort of underlying methods. And there's a sort of demo video recorded by Nina already online if you'd like to again learn how to use the app. And to access this, really the comms team have done a superb job. So you can just sort of search on Google Pf-HaploAtlas and you'll be taken there. So yeah, and then finally on the last slide, we just wanted to show you the Pf-HaploAtlas team. So yeah, well, Chiyun, Nina, we're here already, but Ayub is a data scientist who's done a fantastic job with working on both the front end and the back end. So thank you to Ayub, and also many of you already know Christina, Jacob, and Richard, who've provided us tremendous amount of support throughout the last many months in trying to make this project work. And it's actually so Richard's old code that we used to build the app. So yeah, this project has quite a lot of history actually. So big thank you to the whole team here. And again, thank you to the whole Malaria community for allowing things like PF7, PF8, and all these different data releases to happen. And another thanks goes out to people who helped us launch the app. So the QA testers, the comms team, partner engagement team, the DevOps team, all at the GSU, and also Alfred and Oliver. So yeah, thank you from us all, and happy to take questions at this stage now.
Okay, thank you so much, Nina and Chiyun. Really great presentation and demonstration, very engaging and easy to follow. So well done to the both of you, and thank you so much for your time. We now have time for Q&A before we move on to the demonstration side as well. And just to let you know, John Farrow is busy sharing links to everything that Nina and Chiyun mentioned. So the preprint, the demo video, the app itself. So please do click on those links or copy them onto your computer so that later on, after today's presentation, you can visit them. But also you can visit the Malaria Gen website and social media as well, where those links exist as well. Okay, so time for Q&A. Please either type in the Q&A box or chat box or raise your hand and we'll allow you to unmute to ask your question. We're going to have about 10 minutes for Q&A before we move on. I'm seeing some questions come through. So I will be selfish and start with my own before I read the other people's questions, because I'm holding the mic. So thank you so much for your presentation. As I said, really brilliant and engaging. And I like the iterative nature of it, the fact that you're asking the audience, participants, and users to really give feedback and say, how do they feel? What are they using it for? What do they think should be added, etc. I have two questions. So one is, whenever I hear the word app, I think of my phone. So can I download the HaploAtlas app on my phone?
Yeah, that's a good question. So yeah, perhaps we should specify and call it a web app, but this is an idea that we've been having for a while too. Yeah, we'd love to turn it mobile as well, but you know, we have a huge list of different features and not sure which ones to prioritize. So yeah, if you would like to see a web app, probably yeah, do fill out a form or for requesting a feature, or make a issue on the GitHub page, and yeah, we'll see if we can prioritize that.
Okay, thank you. So that's my first feedback and question, I guess. And to Nina, during your demonstration, you mentioned the fact that some samples were filtered out. So I just want to know, is there any impact on the usability on the app in any way if some sort of data was excluded?
So yeah, that's a good question. For some genes, in particular cases, there are instances where, for example, they may have missing genotype calls across all of the samples, and in those cases, those genes wouldn't be displayed. But that's very few cases.
Okay, thank you so much, Nina. So I'm going to move on to the audience questions. There's about 2 in the Q&A, and as I said, please go ahead and add your questions. There's a question from John Farrow, who says, is there something special about 25 as the default number for the minimum number of samples for a haplotype? That's from John Farrow.
I can take that if you want, Chiyun.
Yeah, go ahead, Nina.
Yeah, so I believe in population genetics, often this is a number between 20 and 30 is quite commonly agreed upon as a robust number to make inferences from. So I think there's a paper published which put a number between 20 and 30 as a good number to use. So we often use 25 as a minimum threshold.
Okay, thank you so much. So there was a question from Raphael Okoth, which I think Richard has answered with a link. The question is, could you be having a reference sequence for P. ovale LDH gene? And I don't know if anyone wants to answer it, but Richard is busy sharing links in the chat as well. Chiyun, do you want to take that, or?
Yeah, sure. So I guess it depends exactly what you want to be doing with ovale, because we are using PF7 data where we're only seeing Plasmodium falciparum samples, really. So we won't be able to see actual ovale samples, but if you just want to compare the ovale sequence, then yeah, potentially we could add that in, possibly as a sort of alongside where the lab strains are highlighted, maybe, Nina, if for example, yeah, like 3D7 and this kind of thing.
I think it's a really good point raised about other Plasmodium species, and yeah, we've definitely considered that there's the potential to expand the app to other species as well. So yeah, if this is something you were particularly interested in, then yeah, please go ahead and put it as an issue, as a feature request.
Okay, there's a question from the chat. It's from Odong Brian, who says, I am Odong Brian, a molecular biology student at Makerere University, who is interested in using the app for my research project. Is the app free of charge?
Yeah, it's fully free. Anyone can go on there right now and browse all you want. Yeah, no need to sign up. You just go on and have a browse.
Okay, I hope you had that, Brian. You can use it for free. The next question is from Edward Rovira, who says, are you considering an additional feature to visualize haplotypes across genes? For example, DHFR, DHPS, quintuple mutants. That's from Eduardo Rovira.
Should I take that, Nina?
Yeah, go ahead.
Yeah. So yes, I think that is a feature we've been considering. It would be super interesting to yeah, see this sort of double haplotypes, I suppose, across genes. But yeah, I don't know. We feel there's just so much we can do with the app at this stage. You're going to be bored of us saying this, but yeah, it would be great if you could sort of describe your use case scenario and yeah, request this feature on our contact pages.
Okay, it's all exciting, Chiyun. Thank you so much. And I think possibilities are endless. There's a question from Dr. Richard Pearson, who says, are there any plans for a similar app for other species? For example, Plasmodium vivax.
Yeah, there are. We would really like to do that and expand it, and we know how it would how we would need to do it. Yeah, so we'd really like to expand it to Pf-HaploAtlas and beyond, and Ovale, as just mentioned.
Okay, thank you so much for that. So I think, as we said, please do give your feedback and use cases, engage with the app, engage with Nina, Chiyun, Christina, Jacob, Richard, and the team. Yeah, because I think the possibilities are endless, as I said. There's a question from Leonard Ndwiga, who says, are there any future plans to enable users to submit pre-analyzed data, such as FASTA files?
Quite an interesting idea, actually. Right, Nina?
Yeah, it's not. I'd say that specific idea is not on our backlog currently, but yeah, I definitely see the value in being able to add your own data and see where it sits within the PF7 or PF8 sort of visualizations from the app. Yeah, so again, please submit that as an idea.
Okay, thank you so much. As I said, we have about 5 more minutes for Q&A before we can move on to the demonstration, which I know all of you are excited for. There's a question from Apti, who says, is that accessible to all countries without charge? So I think it's a question about charge still from someone else. Chiyun, do you want to take that?
Sure, yeah. So the app really should be free to anyone sort of on the globe with an internet connection. But yeah, if not for some reason, do contact us and we can try to sort that out. But really, it should be free to anyone with an internet connection.
Okay, thank you, Chiyun. And by contact, do you mean they should contact support at Malaria Gen, or who do they contact if they want to if they have a query?
I guess any way you'd like. Yeah, contacting through Malaria Gen or just on the app. We have Google Forms you can submit where you can just supply a free form sort of message saying like, hi, I'm from here, can't access your app, and then we should be able to get it immediately.
Okay, thanks, Chiyun. There's an interesting question from Maria Luders Makalinau. How do other countries contribute their data to malaria gen to be included in the atlas?
So I'm wondering if either Christina or Richard could point Maria in the direction of the right people to talk to.
Yeah, shall I take that one? Hi, this is Richard. Yeah, thanks for the great question. So I guess there's a couple of answers here. One is, talk to us about becoming a Malaria Gen partner. If you're not already, send an email to support@malariagen.net. The other thing that we have plans for going forwards is to start incorporating any publicly available any publicly available data into future versions. So, for example, from PF9 going forwards. So all that really needs to happen there is for it needs to be whole genome sequence data, obviously, for this app. And if you have Illumina whole genome sequence data and you can make that publicly available, for example, on the European Nucleotide Archive or the SRA, with some minimal sample metadata, so there's a minimum a year and country of collection of each sample, then the intention is for future releases to pull in all of that data, to run it through our mapping, our genotype calling pipelines, and then ultimately all that data will then end up also within this app. But yeah, do please contact us, support@malariagen.net. But yeah, thanks for the question.
Thanks for that, Richard. Much appreciated. So, Maria, I hope that addresses your question. Do get in touch with the team and they'll assist you. We have about 2 more minutes for Q&A before we move on to a demonstration. So are there any pressing questions? Please do type in the chat or Q&A or raise your hand and we'll allow you yourself to unmute so that you can ask a question.
Nina, I wonder if it's worth showing everyone how to access the contact form, since there's been quite a few questions.
Yes, no problem. Okay, I will go back over to the app. So here on the app, we have a sidebar. And if you scroll right down to the bottom, under Contact Us, you can get over to our GitHub page to submit an issue for a feature request. And also there is a Google form as well if you'd like to just give us some general feedback, and that would be greatly appreciated.
Okay, thank you so much for that, Nina. As I said, we have still some time for Q&A, so please do type in the chat. Otherwise, I'm going to kindly ask that we move on to the next session, which is a demonstration. So you were asked in the advertisement of this interesting webinar that you should bring your own gene that you are interested in knowing more about, and Nina and Chiyun will demonstrate through the app. So for now, I would love for this to be more interactive, as it already is. It's been interactive, but I would like for you to speak if you have a gene. So kindly raise your hand, and we'll select you to speak, and there'll be a demonstration that is done. So I'm checking if there are any hands.
While we're waiting, would you like me to showcase another gene?
Yes, please. Go ahead, Nina.
Okay. Yeah, so to give you all a bit of time to think about what genes you might like to look at, we have another example I could show you. So let's go to DHPS, which is involved in resistance to sulfadoxine, part of the SP treatment for malaria. And let's make sure we're looking at 25 samples. So what I spotted, which I think is interesting here, is if we look at this second haplotype, the second most common in DHPS, I noticed that there's a large proportion of samples that are this sort of yellow golden color, so located in East Africa. So let's click on that and look a bit closer. So what I find interesting about this is if we look here, all these red locations are in West Africa, and we see that frequencies of this K540E mutations are pretty low, but then they're really high over in East African countries. So that maybe tells us something about different evolutionary pressures that are occurring either side of the continent there. Potentially something interesting to flag. And we also have moderate low frequencies in Southeast Asia as well.
Actually, while you're there, Nina, could we have a look at the next haplotype as well? That's looks like it's almost exclusively in the eastern part of Southeast Asia.
No problem. Right. So we're clicking here on this third haplotype, and this has two mutations, A581G and K540M. So let's scroll down. Is there anything you'd like to say here, Richard?
So just looking at, I mean, that looks quite interesting. If you look at, Vietnam bin Phuoc, it looks like this haplotype has been increasing from very low to whenever that was, 2008, up to what's the latest frequency here, over 90%. So, and then some other locations, for example, in the one in Laos at the bottom. So it was at 0% back in 2010, 2011, and then it's again gone up. So it does look like that particular haplotype seems to be increasing in frequency in the eastern part of Southeast Asia, which is potentially interesting.
Nice, thanks, Richard. I'm going to reset the app now. Have we had any requests?
Not yet. I don't know if I should ask this, but it's something that's popped into my mind. So as I heard Chiyun say, it's open data, it's free for everyone to use. But if I'm a scientist and I'm publishing a paper, can I use the app or the data on here to write my own paper? Or is there an embargo somehow?
Yes, so it's definitely free for you to use. We just ask that you acknowledge that you've used the app.
Okay, thank you for that.
But yeah, we really we want this app to be a useful tool to facilitate people doing more research and analysis and genomic surveillance.
Thank you.
Looks like we have a suggestion of HRP. Not sure if that's worth having a go.
So I've just posted an answer to that, Chiyun. So HRP2 and HRP3, they're actually considered to not be in the core genome. They're right on the border of the core genome and the subtelomeres. But because of that, I think we don't have either of those genes displayed within the app because they're not core genes. So I mean, what in effect that means, because they're not core genes, when we create the genotypes for the PF7, we essentially, any non-core gene, we fail all of the variants that are found in those genes, because there's much higher error rates in those genes. So this is potentially something we could look at in the future, just to change, for example, the way that variants are filtered out. So I mean, it would be possible to include HRP2 and HRP3 in the future if, for example, we had a filter that we could turn on or off to remove lower quality variants. So yeah, that's certainly something we could do, but unfortunately we can't show that today.
Thanks, Richard. So just to repeat, this is time for demonstration. If you have an idea of a gene that you would like to see, please kindly raise your hand. Helga is asking about another MSP1.
Okay.
That's from the Q&A.
Thank you. MSP1. Let's type that in. Are you happy to say anything about why this gene is of interest to you?
Helga, you can unmute or type in the chat, but I'd encourage you to speak if you can. If you are comfortable, please raise your hand and then we can unmute you. Helga, would you like to sort of expand on why you're interested in the gene?
There's no problem. If not, I can just run through the app. So yeah, for MSP, something maybe interesting to look at here is just 221 samples made it through for analysis. So we had a lot of samples excluded because they had a missing genotype call. So let's see, we have 5 haplotypes with a minimum sample input of 25 again. And we can see that they've got quite 3 of them are predominantly in the western part of Southeast Asia. And then interestingly, we have 2 haplotypes, which one is only found in South America and one is found only in Oceania and Papua New Guinea. So I don't know what we're going to find now, but I'll just click on this second haplotype. You can see a lot of mutations making up this haplotype. You can see the name is very long here. So, yep, on the abacus plot, as expected, not so many samples being put into it. And what we can find here is that we have a few samples collected in Bangladesh and from around a similar time in Myanmar as well, and that the frequencies in Myanmar from the time point we last sampled were higher than in Bangladesh. So thank you for that suggestion.
You know, it might be worth lowering the minimum number of samples. I think for me, MSP1, that's Merozoite surface protein, so I assume has some antigenic properties as evidenced by all these different mutations, which suggests it's, the protein's under a tremendous amount of evolutionary pressure to just keep changing and changing and changing, and so which is why we have all these different mutations. Okay, how's that looking?
I put it down to 1. We get up to around 1,800 samples available for analysis. Oh, okay. But at that point, we exceed the number of haplotypes which we think is sensible to display in the app. So I'm going to change that to...
We try 5, Nina.
Cool. All right. So we've got a few more haplotypes here. Is there anywhere you'd like me to go in particular, Chiyun?
Not really. I kind of feel... I mean, Christina, Richard, feel free to come in here, but I feel like there shouldn't be too many sort of trends for any single haplotype, but happy to be proven wrong.
Yeah, perhaps if I could just jump in here, if that's okay. So I think MSP1, it's a notoriously difficult gene to genotype. There's actually different allelic forms of this gene, some of which are somewhat similar to 3D7, but there are other allelic forms that are so different to 3D7 that reads from those don't even map to 3D7, which is why I think we've got so much missingness in this particular gene. So yeah, there's as well. I mean, we can see here there's an enormous amount of variation, and I think there's a lot more variation, including, for example, indel variation that we're not seeing here. So yeah, a very very difficult gene to look at.
Okay, that's actually, thanks, Richard, for that, because Helga is corroborating what you just said. She's saying the reason why she was interested in the gene is because there was a lot of mutations in the gene when you looked at samples in Benin that were amplified and sequenced. That's from Helga, who had asked for this gene. Thanks, Helga.
I think it's actually also it's a gene for which long read sequencing, for example, using Oxford Nanopore or PacBio, is likely to be much better at really understanding the full variation in that gene rather than the short. So this data is largely based on 150 base pair Illumina reads, whereas with the long read you can really get the whole gene within a read. So yeah, thanks for that, Helga.
Okay, we have about 9 minutes to go before the end of this very interactive webinar. So please do raise your hand or type in the Q&A or chat box about which gene you'd like to be demonstrated. And Nina and Chiyun can go ahead and demonstrate for us. So there's still time. I encourage you to, there's a comment or question in the chat. What are the latest findings from Malaria Gen regarding the genetic markers associated with drug resistance in Plasmodium falciparum? That's from UPT.
Yeah, should we maybe look at like DHFR? Would that be a good one maybe, or possibly CRT?
Yep.
See what patterns we can spot there. We're going with DHFR. So I believe the mutation that confers pyrimethamine resistance would be S108N apparently, which is a whole load of them. So
yeah. And
I guess any of the ones that include S108N, I suppose.
we have the triple mutant here.
Yeah, should we try that and see what we see?
Let's see. So there we go. Yeah. So would you like to describe that, Chiyun? I can do that if not.
I can have a first go. So yeah, we can see in Africa this haplotype is very, very prevalent. It's almost 100% in most places. But as we look in Southeast Asia, there are a couple of locations where the haplotype frequency is decreasing with time, and perhaps we can yeah, Richard, I assume this is due to sort of decreasing drug pressure here.
I think what probably is happening there is I'm guessing it might have gone from a triple to a quadruple mutant. I think one thing that is interesting here is if you look at some of the older data, so there's if you look back in the 90s or the 80s in Africa, so there's a couple of time points in Kenya and one in Gambia where, yeah, the frequency of this was much, much lower, which is, you know, which speaks to how useful it is to have, yeah, historical data as well as more contemporary data to be able to see what's happened historically. I mean, yeah, you might, if you look at maybe the next haplotype on the list, well, yeah, the quadruple, I'll guess you'll see that's sort of the opposite picture to what we've just seen. So where it was decreasing before, yeah. So for example, in the western part of Southeast Asia, for example, in Tak province in Thailand, this is has been at very high frequency for some time. I think you were just looking at the Lai in Vietnam, that it looks like the frequencies, yeah, increasing in, well, a couple of places in Vietnam.
Yeah.
So I think, yeah, what's essentially happened is the triple mutation was at pretty high frequency, but that's probably now been largely replaced by the quadruple mutation.
Great, thanks both.
There's been a question from the chat from Malaria. I'm not sure if that's pronounced it, but they talk about HSP70 as a gene for us to study in the chat, and later on KDR. Should we start with HSP70, Nina?
Yep, HSP70. Let's have a look. I don't think I've looked at this one myself yet. So, okay, so with 25 samples, we can see 2 haplotypes. Should I lower the threshold, see if we change that a bit? Okay, so just to say as well, we have 11,000, just over 11,000 samples making it through to analysis here. So yeah, not a very diverse gene based on PF7 data. Most samples have the wild type haplotype with no mutations, so no dots here. The next most common exclusively found in African populations, more so in East Africa, some in Central Africa, and some in West Africa as well. And that's with the D603E mutation. Damilari.
So they're quite interested in like protein-protein interactions with, sorry, I wasn't quite sure if it's to do with the insecticide or the KDR gene, but based on that, I think it's quite interesting how little diversity there is here. So not sure that would mean that, I suppose, that there might be less possibility that any sort of resistance to, or like sort of evolutionary drift towards or away from this protein-protein interaction happening might occur.
Shall I check out KDR as well?
I think KDR is actually an anopheles gene.
Ah,
I see.
So this
Okay.
is just
Got it.
Plasmodium data. So.
Yeah. I wouldn't imagine heat shock proteins would have that much diversity personally. Thanks for your contribution, Damilare.
Yeah, thank you.
Okay, thank you so much for that. Damilare, yes, go ahead and speak. I think you can unmute yourself.
All right, can you hear me?
Yes,
Sorry.
we can hear you, Damilare. Go ahead.
I'm actually asking the questions about the interaction between the HSP70 gene and as well as the KDR gene. And the KDR gene is mostly found is a knockdown resistant gene that has been, I think, from Anopheles. I think one of the facilitator mentioned that about the Anopheles, but I'm not so sure. So I want to check out the protein-protein interaction between the HSP70 gene and it can interact with the KDR gene and possible outcome, seeing that if these two can interact, can be used to profound or to propel, to come out with another active ingredient in order to combat this Anopheles gambiae lactose. And also, I'm trying to look at also if this haplotype offset can be carried out for maybe a KDR gene as well. Can I see that? So.
So yeah, if KDR is an anopheles gene, currently Pf-HaploAtlas just deals with Plasmodium falciparum genes. We do believe that the sort of architecture of this app could, in theory, be applied to other organisms, but currently it's not. So unfortunately, Pf-HaploAtlas couldn't help you with KDR, but it can with HSP70.
Okay, so what you're saying now is that KDR gene won't go with HSP70, right? But only with PFMDR.
What was the last? Was the last?
Is that what she's? Sorry.
Sorry, I didn't catch that. What were you saying?
No, what I'm trying to say is that the protein-protein interaction between a KDR gene. A KDR gene is a lockdown gene that is used maybe like using a parasite rod insecticide to, and now we discover that there is an resistance. The mosquito have been able to develop resistance towards the same insecticide. And that's the particular gene, KDR gene, knockdown resistant gene. So checking the knockdown resistant gene together with the maybe an HSP70 gene to maybe if they can bind, checking the protein-protein interaction between the two, that could help him. I just want to know if it is very possible. That's why I'm raising this kind of question here. If it is something possible. Or what do you think?
Yeah, well, really, we're focusing on sort of the malaria genome here, but as Nina mentioned, we could make a similar app for the sort of anopheles genome. However, in terms of the exact protein-protein interaction, you'll need another link between, the HSP70 haplotypes that we're seeing here with the KDR gene. It's not that the HaploAtlas isn't sufficient to determine the protein-protein interactions.
Okay.
But I think I can imagine ways of bringing in like protein-protein interaction data from external sources and sort of bootstrapping that into the Pf-HaploAtlas. So if you do feel that's a useful feature, do let us know. And happy to continue this sort of conversation offline as well.
Oh, definitely. Of course, definitely. Yes, we can. So because I'm trying to look into that one. Thank you for that questions. And also, can I ask the last question, sir? I'm trying to look at, can she help me with the haplotype for the KDR gene? I want to see the samples and if there is a white type and also those if there's any variation in those genes. Can you perform an upload type for KDR gene, please? So I can see.
I believe we can't, I'm afraid, because you mentioned KDR as a mosquito gene, correct?
Yes.
Yes. So far we've only made the haploatlas be able to analyze Plasmodium falciparum genes only.
Okay, I get. Oh, wow. Thank you.
Thank you very much.
Yeah, you're welcome. Yes, you can continue.
Thank you.
Okay. Thank you so much for that interaction. I'm going to jump in and be someone who's going to stop your learning and fun. I apologize, but we've reached time and have even gone over time. And I see there's another question, but I don't think we have time to engage on that right now. Please, again, engage with the app. Watch the video on YouTube about the demonstration. Watch the Journal Club recording later. Share it with your friends. Go onto the app, engage with it, learn. Respond on the GitHub link, which is Nina showed you at the bottom of as well as the Google form, where you can give feedback, because as they said, this is iterative. It's about learning from users and people that are actually engaging in public health and scientists as well. So please take the conversation offline. Go onto the Malaria Journal website as well, where most of the information is going to be as well. And I just want to end by reading a very nice comment from Abti Sharafu. He or she said, Abti said, I would like to extend my deepest appreciation to the developers and researchers at Malaria Gen. Your dedicated efforts in advancing the understanding of malaria genomics are truly commendable. The comprehensive data sets and insightful findings you provide are invaluable to the global fight against malaria. Your work not only enhances scientific knowledge, but also paves the way for new strategies in combating this devastating disease. Thank you for an unwavering commitment and groundbreaking contributions to public health. I think that's a very nice way to end it. Just showing you the impact of your work, Nina, Chiyun, Christina, Richard, Jacob, Ayub, and the whole Malaria Gen team, which is a network made up of so many people. And yeah, I think we can end malaria in our lifetime if we continue work like this. I don't know if there are any comments or last words from Nina and Chiyun before I wrap up. Nina?
No, just thank you everyone for your contributions and questions.
Chiyun?
Yes, thank you everyone for being here. Yeah, as we mentioned, it's so nice that we finally be able to share the Pf-HaploAtlas with everyone. And thank you.
Thank you. Richard and Christina, any words from you?
Nothing from me. Thank you, everyone.
Thank you, Pablo, and thank you very much for the excellent presentation, Chiyun and Nina. I would just like to remember everybody that this is made for the community, so please use it always, free of charge, and it's a great resource for all of you. So the intention is for you to use as much as you want every day, all day. And remember, it's free, open source for you.
Thanks, Christina.
Thank you.
Use it, and give feedback as well.
Exactly.
Okay, so I will just close up and say thank you so much to our presenters. Really, this was interactive, fascinating, interesting, and really accessible. Really grateful for your time. And for those of you that were joining, I just want to let you know we are going to have another Journal Club on the 27th of August, which is going to be a vector-focused one presented by Dr. Kelly Bennett. So yeah, be on the lookout on our social media pages for that as well. To everyone, thank you so much. If you have any queries,